Hi everyone, this is Wildlife Bio Gal, live from the Maumee River. In just a short second here, I'm going to let our guests introduce themselves, but make sure that you start sending any questions in that you have about anything with Great Lakes, Maumee research, lake sturgeon stuff, fair game, lake sturgeon, unionids, any of that stuff. Make sure you can have your questions ready and they'll be more than willing to answer them. If you don't get to watch all of this, make sure that afterwards you check it out I'll put it up on YouTube, I'll put it up on SoundCloud as well. Might be a little later tonight, maybe even tomorrow before I get those up. But if you have any questions afterwards as well, make sure that you tweet me or tweet Jess and we'll answer questions that way. So without further ado, University of Toledo Lab out here live on the Maumee River. So Jess, if you want to introduce yourself, make sure your Twitter handle, you throw that in too, so everyone can contact you.
Absolutely. Thanks for doing this, Nicole. So my name is Jessica Sherman. I'm a PhD candidate at the University of Toledo. You can follow me on Twitter. I'm at Aquamare. And my dissertation project is actually looking at habitat suitability to reintroduce lake sturgeon. And this is kind of a segue of that project. And with me I have some of my fellow grad students who work on projects not at all related to this, but who have so wonderfully volunteered a large amount of their time helping me with my surveys.
Hi, I'm Sara Guiher, also at the University of Toledo. I'm a graduate student and I focus on invasive plants.
And I'm Casey Yanos, also a master's student at UT, and I'm working on modeling trophic transfer in Lake Erie.
So the reason we're out here today is I created a habitat suitability model so that we could help reintroduce lake sturgeon to the Maumee River. They've been extirpated for almost a century, and before we utilize the resources to do streamside rearing and reintroduce them, we wanted to first make sure that they would have enough habitat for both spawning adults and their nursery habitat for the age 0 fish to successfully sustain themselves. And so we created this model. And when it was done, we decided, can this tool be used elsewhere, or is it only specific to lake sturgeon? And so we decided to test the transferability of the model on native freshwater mussels called unionids. And so we're using habitat characteristics like water depth, the substrate composition, and water velocity. And we create a map that looks very similar to this, where we can delineate out between what would be poor, moderate, or good habitat, or areas where we could try to survey first to find the unionids that are in the river. And so what we've been doing is we've selected random sites throughout the river in all 3 habitat categories, and we go out and we survey and we determine, are the unionids present or absent, and if they're present, what is their diversity, density, and abundance. And then later we'll take those numbers back, we'll compare it to the suitability model and determine how well can this model predict if unionids are present or absent. And one of the important parts of this tool is the Maumee River is a very large system, and there isn't a lot of information available on how the unionid populations are, how they're faring in the river. And so with such a large system, it's hard to go out and just start sampling anywhere. You have to have kind of an idea on where do you begin. And so we're hoping that this tool will be something that researchers in the Maumee River or in other large river systems will be able to use to say, okay, we need to go out and find what is the status of our unionid populations. Well, where do we begin? Where are some of the habitats that we should be checking first? And hopefully this model will we'll be able to work it out so that it will accurately predict where you could find them and therefore narrow down the search area for researchers who are trying to determine if they're present or absent.
So if you guys have any questions, feel free to start sending them in. Anything about Great Lakes, awesomeness of the Maumee River, why the ducks are upended right back behind us over there. So go ahead and start sending in those questions. So I guess my question is, you do also some lake sturgeon research. How far inland will they go from the Great Lakes?
They'll actually go pretty far inland. So the Maumee River is about 210 kilometers long. It starts at the confluence at Fort Wayne, Indiana, the confluence of the St. Mary's and the St. Joseph River. And historically, there were accounts of lake sturgeon making it all the way to Fort Wayne, Indiana. Now, there are currently several dams that are impeding their movement. The first dam occurs just 56 river kilometers up. And so it's a low head dam at Grand Rapids, Ohio, and so they can no longer get any further up there, plus they're extirpated from the river. So historically they could have traversed the entire length of the river. When they're reintroduced and they sustain themselves, they'll only be able to go up to Grand Rapids.
Okay. So where do the lake sturgeon in the river like to lay their eggs and spawn?
Not on the bedrock, which we're standing on. So lake sturgeon actually will spawn on substrate very similar to walleye. So they're looking for a cobble gravel mix. The hard rocky substrates that have the interstitial spaces, because their eggs are kind of adhesive and they'll sink into those interstitial spaces. They'll incubate for about 2 weeks, but they need that interstitial space to protect them, one, from predators, but also from being dislodged by velocity before the larval fish can hatch out. So this hard, flat bedrock substrate that we're standing on right now would not be good for them to lay their eggs on, but there's actually an extensive amount of that cobble gravel mix that they'll be looking for available throughout the river. And we also know this not just from the surveys we did, but the Maumee River has a pretty extensive spawning walleye run. They have over 500,000 walleye enter the Maumee every spring to spawn. So that was kind of the initial basis for our project was, well, they have a lot of characteristics and excuse me, requirements similar to walleye. So if the walleye are doing well, we could surmise that the sturgeon would do well.
Great. There was a question a few minutes ago, and unfortunately I was in the middle of asking my question, so if you want to repeat that question again, it was something about the Maumee River, so if you want to resend that in, I'll try to catch it this time. I caught it right as it was disappearing off the screen, so sorry about that. So we always hear about like huge phosphorus loads and everything coming through. Like this seems to be like the major point of, you know, cause of like some of those harmful algal blooms in Lake Erie. So what can you tell us a little bit about that? I mean, I know it's maybe a little bit outside your scope of research, but you know,
it is outside the scope of my research, but I can talk a little bit about it from what I heard from some of my colleagues. So yeah, the Maumee River, it's the largest tributary to the Great Lakes by watershed, and unfortunately, nearly its entire watershed is agricultural based and urban, and so it does get a lot of phosphorus coming from the landscape surrounding it, both in ag and in the urban and industrial areas. And there have been some researchers who have studied the influx of phosphorus and found that yes, the Maumee is the largest contributor to Lake Erie, far more than the Detroit River. And there's not much that we can do about it right now at this point in time, but there's a lot of researchers and a lot of organizations that are out there that are actively working in the watershed. They're working with homeowners and farmers and trying to reduce the phosphorus load, trying to find best management practices. There are some land trust organizations that are trying to put in more buffer strips, more preserves along the river to try to catch that water, filter it out before it enters. So people are actively, they're aware of it, they're trying to make it better. It's just, you know, it's an uphill battle for a while. There's a lot of phosphorus in the system, there's a lot retained in the sediment, so it's not something that'll go away anytime soon. But yeah, the Maumee is a pretty big contributor for the Great Lakes, especially Lake Erie.
Okay. The question we had was from Science in Michigan. It's how high is the Maumee at this time of year, and how is that affecting your models?
That's a really great question. So one of the issues with working in the Maumee system is that it is incredibly flashy. And right now our flow is about 800 cubic feet per second. It's a little bit higher now because we had quite a bit of rain at the tail end of last week and over the weekend, so it's come up a bit. A few weeks ago, this part of the river was actually dewatered. And so that is one of the things that we do take into consideration with the model is the timing. And so for instance, when we build the model for the lake sturgeon, when we do the habitat model for the adults, we use an April discharge. When we do the habitat model for the age 0 fish, we use a June discharge to kind of capture the difference between the fluctuations. And so it has a lot to do with, you know, if that habitat would be suitable or not. And one of the things we have to consider is just because the substrate is perfect and the perceived velocity in the model would be good, if we come out here in the summer and it is bone dry, you know, it's not an area that we're going to find unionids, and it's not an area that would be good habitat for the age 0 sturgeon who stay in the river for the whole summer. And so it does affect how our model operates.
So how do you guys go about sampling for unionids? Can you like demonstrate real quick, or is that something that's really easy to do?
We can do a little bit. So what we're looking at is we set up we'll set up a 10 by 10 meter quadrat.
Yeah, I was gonna say, make sure I don't step off as I'm trying to record you here.
We would set up a 10 by 10 meter quadrat, and it's a lot easier to show in some of the softer substrates, but we'll cordon it off, and it's all a tactile search. So in the wadable parts of the stream, and part of our model validation is that we're strictly working in the wadable parts of the stream. So everything that would be chest deep on us and lower than that. And so if it's too high, we actually have clam rakes that we're using, and we'll kind of rake the whole area and determine if we can pick them up or not. But otherwise, you know, we have this area, we're working in groups of 3 or more people, and we actually just get down and we do tactile search. If it's a sand gravel substrate, I mean, we're picking it up, we're sifting it through our hands, we're spending some time moving it around. We'll do the whole 10 by 10 quadrat, and we usually have people that kind of go up and down, we have people that zigzag, we have people that are all over the place, and as long as we're finding mussels, we keep our survey going for 4 additional minutes. So after 4 minutes from the last time a mussel had been found by the whole group, that's usually about the time that we would determine that the site has been exhausted. Okay? So it's all tactile searches where we can, and using the clam rakes when the water's too deep.
Okay. So you guys have any more questions? Keep sending those in. Oh no, you're good. So this is all live and fun. That's the best thing about, you know, Periscope, is we get to have, it's just guerrilla broadcast that we get to go out and do. Like, you know, hopefully we'll see some of those great blue herons flying through that we saw just a few minutes ago. They're just downriver there. I don't know. So question for Fame Woman: have detection methods evolved at all over time?
Detection methods for the unionids?
I believe that's what she's probably asking about. See if I can get clarification here real quick, but I'll assume unionids.
So, and the detection methods are a little bit different based on what you're looking for. So some researchers actually go out and they will excavate down to a foot, and they'll take areas that are about 1 meter square and they excavate the entire meter, and they're sifting out because they're actually looking for the very small juveniles that are burrowed in the substrate. We're not quite looking for the juveniles. We have found some during our surveys, and we can with our survey, but it's not a thorough review of if the juveniles are present or not. What we're looking for are the adults. And so the survey methods have evolved over time, depending on what you're looking for and what system you're in. So I think a lot of researchers initially were using things like the Ponar grabs, you know, before the scuba equipment was available. And in a riverine system, there's several methods that involve, you know, finding a unionid first, putting a stake down, and then doing, you know, a line transect around that. And that's kind of under the assumption that because you usually see them in communities, that one unionid would predict more unionids that are there. We're not quite doing that type of methods. We randomly select a site and determine if they're present or absent.
Another question we had was about the water. So is it safe to touch, or do you have to use gloves?
That's actually a really good question.
Yeah, we joke about it, but it is. I mean, it's a legit question.
The microcystis algae that blooms out in Lake Erie, when it's blooming at its highest max capacity, it's releasing a toxin called microcystin that can actually be an irritant for the skin, and obviously you can't drink it as well. Out here you do see some microcystin blooms in the river, but because it's a flowing environment, it doesn't persist and isn't as high a population as you would see out in the lake. So we don't use gloves. We're usually in it, you know, sometimes chest deep in these areas. And so far so good, you know, we haven't had any skin irritation. Some people can experience it if they have sensitive skin, but it's not something we're too worried about. You know, we try not to drink the water. Try not to get it in our mouths.
Another question was, do you see any snakes out here?
Yes, we have northern water snakes. So we found them last weekend, a couple of them. We were doing our surveys and found some juveniles along the shore. And then, because we had a group of volunteers out last Saturday, we got a fish net, a seine out, because we just wanted to seine the river and give them a little bit more of an example of the ecological integrity of the river. And so as we were seining, we caught a fairly large northern water snake. So, yeah.
When you're fairly large, like how large is fairly large for a water snake?
So I thought it was about this big.
Okay.
It was probably about this round. I thought it was fairly large. You guys. Compared to the little guy we found the day before.
Oh, okay. So another question was any turtles or anything that you find out here?
Yes, we do see a lot of turtles. Can I defer that one to you because I think you know your turtle species a little bit better?
I'm not sure of any that we've seen specifically here.
Okay.
We saw just those tiny little ones on a log when I was out. So we do see turtles.
We
Okay.
do see
Not sure what they
turtles.
were.
They're probably paint turtles and map turtles. But in this area, we do have Blanding's turtles, and then the more terrestrial, we do have box turtles as well. And I'm sure
Okay.
there's a few other species, but I'm not a turtle expert. But we do see on the tree stumps and fallen logs little turtles.
Okay, so we had a quick question if we're in Michigan. No, we're just across the border in Maumee, Ohio. And then the question coming in, what's the difference between the adults and juveniles when it comes to, oh, darn it, I've missed the last end of it. Science of Michigan, if you could please resend it, because I'm pretty sure that was you who sent that out, and I know you'll be good at sending that, so. What other questions I have? As soon as Science of Michigan will send it, because I know it's probably a pretty in-depth question knowing him. But really simple question. So if this is kind of more of like one of the lower points, is this usually the low time of the year for the river?
This is usually the lowest time of the year. Now, like I said, we had some pretty extensive rain over the weekend, which has brought it up a bit. But yeah, this is August, September are usually the lower points in the year. Last year was an anomaly. That was something we had to really consider when we were creating our map, was that last year we had so much rain, and that's when we did a lot of our surveys for the sonar and our water depth. And so we actually had to deflate those water levels to get them to be more accurate of the year-to-year surveys.
Okay, I've got a quick question followed by the more extensive question. So
Yes.
are lake sturgeon native to Ohio and this river?
Yes, lake
Okay.
sturgeon are a native species. You find them all through the Great Lakes Basin, the Mississippi Drainage, and the Hudson River Basin. They've been extirpated, which means that they are gone from the river, and they've been extirpated since the late 1800s, but they are native.
Okay. And then the question from Science in Michigan, if I remember reading it right, was what is the difference between the adults and juveniles you'll find? So I think they're asking like where maybe you'll find them in the river, and maybe I think that's
Would
probably...
that be the lake sturgeon or the
sorry,
unionids?
the unionids.
The
yes.
unionids. So what is the difference between where you would find them?
Maybe.
You likely find them in fairly similar habitats. What's interesting about unionids is that they have an obligate host. They use fish as an obligate host for their larvae. And so as a unionid spawns, they create this lure or a sac that has their larvae in it, the fertilized eggs. And fish are usually attracted to the lures because some of them mimic prey item, and some of them are just kind of this broadcast net that a fish swims into. And so anyway, a fish will come in contact with the unioniid lure, and they get these glochidia that will clamp down on their gills. And the glochidia stay on the gills of the fish for usually 2 to 4 weeks, depending on the species and depending on water temperature and some other characteristics, before they drop off. And so oftentimes the unionids, the larvae, are dropped off in areas that can be quite a distance from the adults. But what most unionids are looking for in general is a substrate that they can burrow into. So anything that is too rocky or anything like this bedrock flat, they're not going to be able to burrow into. And they need to burrow so they can sit upright in the water column and filter feed. And so if a glochidia is dropped off in an area that is not suitable, it won't survive. So typically you would find that the larval unionids or the juvenile unionids and the adults are occupying very similar habitats, but because of that intermediate stage where they're on the fish, you don't always see them in the same area. And the other part of that is that, I kind of just lost my train of thought. Oh, the juvenile unionids are usually much deeper in the sediment, so they're not filter feeding. They call it a pedal feeding. They're just using their little foot as they're burrowed in the substrate, and they stay burrowed for usually a year or two before they emerge and sit upright in that substrate. And so that's kind of the other difference too.
Okay. One question we had was, no, unfortunately, someone was asking if we had any unionids on hand, but unfortunately they haven't been able to catch any out here at this point today. Yeah, maybe for a future broadcast we'll be able to get some. But one of the questions was, what kind of fish species do you see out here, and would you see any of them now, especially with it being so low? I'm adding on to the question that they asked.
Yeah, so I'm gonna segue real quick. So what we're seeing right here, this is actually Bluegrass Island in the middle of the Maumee River. And so this is one arm of the river as it diverges around the island. On the other side, it is much deeper. We actually can't wade across it. And so on this side of the river, it's obviously more shallow. The kind of fish species we see, there's quite a few bass species. There's white perch, there's a lot of catfish, channel cats, flathead. Upstream further, we see a lot more of the mad toms and stone cats. There's also quite a few minnows. We have some darters, greenside darters, Johnny darters, log perch, freshwater drum, and gizzard shad. And I'm sure I'm missing a few. So largemouth and smallmouth bass. People on the other side of the river, there's a few deeper holes, and you see them fishing right now. They'll be fishing for the bass, and then upstream a little bit, you see them fishing for the catfish quite a bit. The walleye are mostly migratory. I have been a little bit upstream, but in this part, we usually see that the walleye will migrate back out into the river. So you don't see a lot of people fishing right now for walleye in the river.
Okay, one of the questions too was, do you have a home tank? And so I'm wondering if they're asking, like, do you have a tank, back in your lab of where you have unionids, maybe that you could show off later, put a picture up on Twitter of them so people can see what they look like.
Unionid beds that are upstream of here, so we can definitely put those pictures on Twitter later so you guys can see what those look like and see we've got some really cool species out in the river.
Cool. Well, with that, you want to wrap things up?
Yeah.
I know you said you had to get out of here. She's got a meeting to go to, so we snagged her along here.
I have to shower first.
Yeah. So.
Yeah. Thanks everyone for tuning in. I really appreciate it. And like Nicole said earlier, if you have more questions, we are more than happy to talk to you about them. So you can send them to Nicole. You can send them to me. I'm at Aqua Amare, and I'd be gladly answer any unionid and lake sturgeon questions you have.
All right, I'm going to say thank you to everyone from University of Toledo for coming out and letting us broadcast here. A couple quick wrap-up things. Just everyone, if you have questions, obviously get a hold of me at Wildlife BioGal. I'll put everything up on YouTube and SoundCloud later. Also on Friday and Saturday this week, I'll be up at Sleeping Bear Dunes doing some broadcast up there. We'll experiment with some Facebook Live as well, so if you are on Facebook, check me out. It's at Wildlife BioGal on Facebook, and I'll get the times and stuff probably either the day of or maybe even 15 minutes before we're going to broadcast with those, because we're doing a BioBlitz up there with the Cerulean Center, so it should be really cool. I mean, Sleeping Bear Dunes, I mean, anyone from Great Lakes area knows how awesome Sleeping Bear Dunes is, so
Fantastic.
We'll be out there. Yes, definitely. We'll be out there live broadcasting, so make sure you tune in then. And other than that, see you guys later, and have a great afternoon. Bye.